Chapter 12, Specimen Handling, Transportation, and Processing. In this chapter, we're going to describe at least 3 sources of pre-examination errors that occur during blood specimen handling. Describe at least 3 sources of pre-examination errors that occurs during blood specimen transportation. Describe at least 3 sources of pre-examination errors that occur during the specimen processing or storage. Name the 3 methods commonly used to transport specimens, and describe at least 3 basic shipping requirements for safe transportation of infectious substances. Specimen handling after the venipuncture. Remember the phases in the laboratory cycle as presented in Chapter 1. The pre-examination pre-analytical phases of the initial test request, specimen collection, examination, analytical phase, in the final reporting of results. Please remember that the pre-examination pre-analytical phase is part of the phlebotomy's duties. TAT in your textbook on page 397 is the time it takes for a report is being also referred to as turnaround time. It is the time that it takes to order, collect, transport, process, and analyze, and a result reported for a lab specimen, as shown here. Blood and other specimens must always be delivered as quickly and safely as possible without necessary delays, because this could impact the turnaround time. So routine lab, generally, it takes about 2 to 3 days to get results, where a specimen that's stat, we want to do the whole process from ordering, collecting, transport, process, analyze, and results reported within 1 hour. There's different patient variables. So identity, age, gender, pregnancy, medications, fasting versus non-fasting, diurnal variations and refusal to cooperate, patients unavailable, stress and anxiety could impact our ability to obtain a sample. Transportation handling variables, so the correct filling, mixing, tube leakage, breakage, and shaking also could impact the pre-examination variables and the integrity of our sample. And this can be found on page 397 and 398 in your textbook. Specimen processing and storage variables, centrifugation, sample registration, distribution, processing delays, and contamination of the specimen, exposure to heat and light depending upon the lab test. Specimen variables, hemolysis, which is the breaking apart of the red blood cells, blood volume in the tube, mixing of anticoagulants. We want to make sure that when mixing the specimen by gentle inversions is important when the tubes contain an additive to ensure even distribution of the additive into the blood. If they're not mixed adequately, tiny fibrin strands or clots may form within the specimen tube and clog instruments, leading to erroneous test results. Mixing the specimen, again, gentle inversions, even distribution of an additive avoids improper clotting or hemolysis. This shows the proper representation of aligning the label. It can be found on page 399 in your textbook. Labeling the specimen correctly involves 3 steps. First, the label should be placed on the tube immediately after collection without delay. It should be oriented correctly on the tube. The label should include the patient's first, last name, identification number, date and time of collection, the name and initials, the identification number, or birth date, the person collecting the specimen, and other optional information, including the test ordered, volume, and physician's name. Many labels are compatible with laboratory instruments, but please be aware that you need to sign or initial each labeled specimen according to the instrument. And third, whenever possible, the label should be compared to the patient's identification information. We need to make sure it's properly aligned. Also, too, that it's not overlapping with our tube stopper cap. This next picture just shows the technician labeling the sample, adding their initials and time, and then verifying with the patient, which is important. Specimen handling procedures, transportation to the appropriate lab as soon as possible. We're going to place our samples in these biohazard bags, and then we will mark the transportation temperature, whether it's freeze, refrigerator, room temp, is it stat. And then sometimes we have these labels to indicate to our lab, you know, fax results. Is this a stat test? Is it from the outpatient center? So that they know where it came from. So the transportation to samples could occur as soon as possible. Special handling procedures are needed for specific laboratory tests, cooling to slow metabolic processes, transport at body temperature, and protect from light. Your table in 12.1 is really essential to cover. So specimens that are thermoable or may require chilling means the substance is sensitive to higher temperatures, and the specimen being tested for these need to be chilled. This is done by placing the specimen in an icy slurry, and the specimen needs to be tested within 2 hours of collection. Laboratory test examples include ammonia, blood gases, gastrin, lactic acid, parathyroid hormone, and pyruvate. Specimens for electrolytes should not be chilled. As we continue in this table on page 401, specimens that require 37C, these specimens require either a commercially available heat block, as shown here, or a heating block for transportation and handling purposes. The tests are cold agglutinin, cryofibrinogen, and cryoglobulin. It's really essential. We will warm the tube prior to collection, and then we'll put it back on the warmer once the collection is done. Our third specimen handling requirement is specimens that are photosensitive or should be protected from light. Photosensitive means that the substance is sensitive to light and will decompose after exposure to bright light. So it's really essential that when we collect these, that we protect it from light, either with an amber brown biohazard bag, an aluminum foil wrapping around the tube, or transportation container that does not allow light to enter. Some common lab tests is bilirubin, beta-carotene, folate, vitamin A, and vitamin B6. It's really essential that we make sure that we protect these specimens because, again, as they are exposed to light, they will start decomposing. And this shows the image of using foil to cover these samples. There's also plasma-based coagulation analysis, and this is dependent upon the lab, but it gives a specific time frame of testing for coagulation testing. And the use of ice or ice packs is not recommended because the possible cold agglutination of clotting factors needs to be assessed. So please make sure to view this chart on page 401. Microbiological specimens transport as soon as possible to the lab so the specimen can be transferred to culture media or analyzed. Blood culture is usually collected directly into the culture media. They minimize the possible contamination and speed contact with the culture media. It is really essential that when we collect these, blood culture specimens are usually collected directly into culture media, which again minimizes possible contamination, speeds contact. Since they provide nutritional ingredients for bacterial growth, the sooner they are incubated, the sooner the bacteria will grow, and the sooner a positive result can be detected. So specimens are placed in an incubator. Generally, blood cultures take about 3 days to determine if the patient has a positive test result. Specimen transportation from remote sites, found on page 403. From ambulatory clinics, home health collections, follow safe handling guidelines and standard precautions, and use enclosed or lockable containers to avoid spills in the car. It's really essential that we keep warmers, and cold packs could be used in different conditions or weather. In general, the rule of thumb is that blood specimens for chemistry tests that require overnight shipping should be centrifuged within 2 hours of collection, and the serum or plasma should be removed from contact from the cells to provide longer stability of the sample. We're going to use biohazard warning labels on the containers with notification procedures in case of an accident. Use cold packs during hot weather and heat source in freezing weather to avoid the specimens getting overheated or freezing. Specimen delivery methods. So we have a couple different type of delivery methods. There is the hand delivery. Hand delivery is required for timeless of delivery to laboratory. A log or data sheet for the specimen that are hand delivered to the laboratory is commonly used. And these are placed in test tube racks and then awaiting delivery to the lab. Then there's the pneumatic tube system. It's a shock absorbent padding and liner. The tubes are separated from one another to prevent spillage or breakage. They are placed in a biohazard bag and then in a plastic sleeve, and then they're transported automatically through the hospital. Please be aware that specimens for coagulation testing should be protected from vibration and checked to avoid platelet aggregation during transport. And also, too, we don't want to place anything in this container that is what's called unretrievable. So anything that's invasive, so cerebrospinal fluid, biopsies, things that generally would you need a provider, or if the patient's blood sample was really hard to collect, we'll want to hand deliver those as opposed to using the pneumatic tube system. Because if the specimen were to get lost, we may not be able to retrieve it again. Transport via automated carrier. They provide sometimes motorized, computerized, or resemble small elevator-like systems. The delivery of specimens can occur by these means in small container or cart attached to a network of track that is routed to the appropriate site. It's just another way. There's also transportation by drone. Commercially delivery applications using drones are being developed. Preliminary reports from Johns Hopkins suggest that blood samples for routine tests and blood product delivery can be maintained at safe temperature, and these would be ideal for remote sites. Strict regulations from the federal agencies and applications in remote, rural, or impoverished locations during national disasters or in active war locations. This would be a good way to get blood products to a patient as well. So processing steps on box 12.1, starting on page 407, it covers pre-centrifugation. So this is after the specimen is collected, but before centrifugation. Then there's centrifugation while specimen is in the centrifuge, and post-centrifugation after centrifugation of the specimen, but before removal of an anti-aliquot or serum or plasma for testing. Please be aware that I would like you to review this in detail, as you will have a lab on specimen processing and understanding the requirements for each step. Okay, as we review this chart / table, it's really good to learn the concepts of how to properly balance the centrifuge, and also read any of their clinical alerts present. Noteworthy features on figure 1210 we will review in a couple slides. CLSI recommends guidelines for handling and processing. Some do not require centrifugation, some hematology and coagulation tests, and they need gentle mixing using a mixing tray, but are not required to go in the centrifuge. Pre-centrifugation, the specimen handling processing after collection and prior to centrifugation, which is the phlebotomist's responsibility. For specimens that do not require centrifugation, the rule of thumb is the serum or plasma should be removed from the cells as soon as possible and not exceed 2 hours from the time of collection, especially if we're waiting for a carrier service to pick up the sample. Plasma specimens that can be centrifuged immediately. Specimens without anticoagulant additive, such as our red and SST gold, must clot before centrifugation, and this takes about 30 to 60 minutes. Pre-centrifugation clotting time can be delayed by patient's anticoagulant medication, Coumadin, warfarin, and heparin, and chilling the specimen will be delay clotting as well. I provided a video which I'll share in Blackboard, which shows what snake venom does to the clotting process inside the body. Pre-centrifugation clotting may be accelerated by activators such as glass or silica particles, thrombin, snake venom. They keep photosensitive analytes wrapped in aluminum foil or placed in an amber specimen container. Again, this is just a summary review. What we talked about, serum samples should clot before centrifugation. It takes 30 to 60 minutes. We're going to leave tube caps on during centrifugation. We want to make sure to balance the centrifuge. And in your book, it goes into great detail in an image, and I provided a video as well on how to properly balance the centrifuge. Do not open the top of the centrifuge until it is stopped completely. There is temperature-controlled centrifuges for our frozen and our refrigerated samples. Do not centrifuge specimens more than once, and follow manufacturer directions for use of gel and non-gel devices. We will balance the centrifuge with tubes directly across from each other, never next to each other. In an unbalanced centrifuge, will shake violently if this is not completed. Post-centrifugation, remember to separate serum and plasma from cells as soon as possible. Refrigerate a free specimen accordingly. Specimens may be divided into numerous aliquot tubes and consider storage temperatures depending upon which tests are ordered. Post-centrifugation, we do not want to repeatedly freeze, thaw, and refreeze specimens. Serum and plasma may be left in contact with a gel barrier, separator device, or plunger, and inspect tubes after centrifugation, making sure that we note any changes to the color of the serum or plasma, as shown here in Figure B. This would be specimen was centrifuged properly, but there is slight hemolysis. Specimen A here, the specimen was noteworthy features. First, it was not balanced when it was centrifuged, so this is why it's angled, as shown here. And it also, the mistake has often led to leakage of the specimen into the plasma. Second, the specimen is lipemic, milky, and cloudy, which indicates a high level of lipids in the blood, which means that the patient may not be adhering to the fasting diet. Image C. This specimen does not contain a barrier, but notice how it separates into the cells and plasma. And then D, the specimen is grossly hemolyzed. The plasma portion is hardly distinguishable from the cellular portion because of the deep red hemolysis. It almost makes the specimen look like it's not centrifuged. This was likely the result of excessive mixing and irrigation, so things we note after we take the specimen out of the centrifuge. We want to store tubes in an upright position with secure closures. Excessive aggregation may cause hemolysis or breaking apart of the red blood cells, and specimens may also appear lipemic, which is a milky, cloudy appearance. Consider the following factors using the right centrifuge: the type of tubes, rotator, spin time, speed, and capacity. In your book, this goes into great detail. We need to be aware of tube size, also to if we're running 100 samples, we don't want a centrifuge that will only take 6 tubes. We'll need one that's larger. Considering the following factors: so temperature control, noise level, proximity to other instruments, maintenance and training for centrifuge operation also needs to be taken into consideration. Primary source of shipping biohazard specimens, although not commonly done, phlebotomists may not have exposure to this. It is something that we need to be aware of. Primary source for regulatory governing specimens and biological shipments in the United States. DOT collaborated with WHO and the Union Committee on Experts and CDC to establish these guidelines. Every lab has a different policy and procedure when it comes to shipping biohazard specimens, so you will have to reference to that based on the lab you are working in. So infectious substances, known or reasonably expected to contain a pathogen. So pathogen would be a bacteria, virus, fungus, parasite, infectious particles that causes disease in humans or animals. There's two categories of infectious substances. Category A, exposure is capable of permanent disability or life-threatening. Category B, exposure is not usually life-threatening or capable of permanent disability. We're going to train all employees involved in transport materials must meet strength, absorbency, and requirements. Identify, classify, pack, mark, label, and document according to procedures. Reporting laboratory results, there's written reports. We're going to confirm date accompanied by permanent reports available in laboratory and patient's medical records, must contain ID of the patient, location, and physician, date and time of specimen collection, and description, source of the sample, and labeled precautions. CAP has suggested the healthcare personnel considering the following data for laboratory tests. Found on page 417, please review these 11 items. For paper copies, compactness and ease of shipment mailing, consistency and formatting is key, clear understandability of instructions or orders, and logical location of the patient's medical records for reference of laboratory reports. With written reports, there's sequential order and multiple results on a single specimen. Listing of reference ranges or normal, abnormal, critical values, these might need to be called to the physician as opposed to just sending the report. Assurance of accuracy, requests of transcripts, and administrative and recording record keeping value. So reporting laboratory results, verbal reports. The use of verbal and telephone reports has declined because of easier, more reliable computer access and concern for privacy. Verbal reports, although useful for reporting stat results and critical values, are more prone to error because of miscommunication. So it is essential that when you're doing a verbal report, you fill in the required documents as shown in this image here. The documents will have the following information. It will need the patient's name, ID number, person receiving the report, date and time, information given the person issuing the report, so that way we have a secondary resource to reference back to in case of any miscommunication. Please also read the clinical alert on page 418. Reporting laboratory results, we have computerized reports now, rapid online report system, which is really nice. It can become available to any physician, more reliable than verbal reports. Hospitals may have computers located in each patient unit, and the results are immediately displayed in each patient unit. It allows for collaborative patient care as well as privacy. And the other thing too, it's more reliable than verbal reports. Generally, a verbal report will be given via phone, and then the computerized report is also followed up to the physician as well. It's really essential that in Chapter 12, you review that box 12/1, which covers the specimen processing application. We will have a lab on this, and also understanding specimen handling after the venipuncture and the criteria that could change based on the sample received. This concludes Chapter 12's narrated PowerPoint. Please make sure to review the study questions, case studies, and action and practice to prepare for your tests later on this week.